This is the picture that I prefer to use to talk about the different structures of the eyeball. And in the lecture handout or in the PowerPoint presentation, you'll see the information listed in one way. But when I actually do a presentation on the eyeball, I like to use the path of the light to direct my commentary about the different structures. So out here is your open environment where the light is coming in, and then we have the different layers of the eyeball to go through and talk about the different components. So we're going to start here, and the very first layer that we need to remind ourselves of is what is being detached here and here, and this is the conjunctiva. And the ocular conjunctiva is covering the surface of the eye. The light comes in, it passes through the conjunctiva. It's a single layer of epithelial tissue, so there's not a lot of inhibiting of the light coming in. The next object that we have is going to be the cornea. And the cornea is a piece of transparent tissue that you can see is arced here. And as the light enters this tissue, because the tissue is bent, the light bends. And so this is the area that you have your very first focusing of the light as it's passing into the eye. And this is going to be major focusing. This is a pretty significant bend, and so as the light comes in, there's going to be a fairly significant bend to it. And so this is your major focusing. Let's just leave it at that. Now, as it comes in and bends, it is then going to enter the anterior chamber of the eye. And the anterior chamber of the eye is going to be filled with aqueous humor. So, as the light enters, it then passes through this opening, and this is the pupil. And the pupil is the opening within the iris. The iris is usually the colored veil of tissue. So if you say you have green eyes or blue eyes or brown eyes, it's this tissue here that is going to have that pigment and be the color of your eyes. Well, it's a pigmented tissue because wherever the iris is, you want it to be able to block the light so that the light is only passing through this opening of the pupil. Now, just behind the iris, you're going to have the posterior chamber that is also filled with aqueous humor. So the way this works is right here you have the ciliary body, and the ciliary body is going to be glandular in nature, and it is going to produce aqueous humor. Aqueous humor is then going to flow over the surface of the lens, pass through the pupil, enter the anterior chamber of the eye, and then it is going to go into these little holes here and be reabsorbed and then eventually put back into the venous system. So this little hole is the opening to the scleral venous sinus, and that is a very technical and wonderful name, but I prefer the canal of Schlemm. And I like to really emphasize the schl. So the canal of Schlemm, it reminds me of a schlemiel. I just like the sound of the word. So what you need to think about is that this does actually have a clinical application. So if I somehow block or disrupt the reabsorption of the aqueous humor, then the amount of aqueous humor that it reabsorbs is reduced, and it can go below the amount of aqueous humor being produced. So I get more and more fluid, and it builds up in here, and it pushes on the surface of the eye, and now I have increased intraocular pressure, which is basically the major symptom of glaucoma. And glaucoma can cause death of the retina and cause you to go blind from the outside in, where you develop tunnel vision. And the reason for that is as the pressure is increased, then starting at the periphery, you shut down the blood supply, and the retina will then die in the periphery at first because the blood is not making it to the ends. And then as the pressure gets worse and worse, you get closer and closer, and you get your tunnel vision. So maintaining the health of the canal of Schlemm is an important endeavor to maintaining your visual health. Also, I'll go back and just make this comment. If you didn't realize that the cornea was where you had major focusing of your light, just think about the types of clinical things that we do to the cornea. There is a procedure where I take the ocular conjunctiva, I peel the epithelial layer off from the cornea, I shave the cornea and change its shape to fix a visual defect, and then I put the conjunctiva back over it, and that process is called LASIK surgery, and I can fix nearsightedness and farsightedness and even astigmatism to some extent, and then the person is free of wearing glasses. So changing the shape of the cornea changes your vision, so that certainly means that this is involved in bending the light, and this is where you're going to get your major focusing. While we're here on this again, the attachment of the cornea is then to the outer layer of the eye, and that is going to be your sclera. And it's commonly referred to as the white of the eye because in humans, when we look at it, it's white on the outside. So this is dense connective tissue, and it is the outer periphery or the most superficial layer of the eye. And it's dense and connective tissue-like because this needs to help hold and maintain the shape of the eye. And the shape is directly related to your vision as well because it needs to be a nice globe. If you elongate the eye a little bit, then you get an astigmatism, and the image is actually going to be over here or over here or over here where it's in focus when the major focal point of the light should be there. And so if your eye is out of round or out of shape, then that can affect your vision. And so the sclera does have some sort of clinical application as far as maintaining the shape of the eye. Additionally, you need a strong connective tissue like outer coating of the eye so that you can allow for the attachment of these extrinsic muscles of the eye so that you can move the globe of the eye in the orbit. Now we came down and we were just in the posterior chamber talking about the aqueous humor. So let's go back to that. Here we have, remember, your ciliary body that's going to be producing the aqueous humor. And then associated with the ciliary body, sort of the second half of it, notice you've got some ciliary muscle here. And this muscle is going to be used to change the shape of the lens. And it does that by interacting with the suspensory ligaments of the lens. And as this muscle contracts, it actually pulls the suspensory ligaments up and toward one another. And this allows the lens to be more globule. And the more round the lens is, the better able you are to see close up. And so if you're reading and you have the textbook fairly close to you, then these muscles are going to be being used. They may be contracted for several hours, allowing you to read. And with that, you could get tired eyes. And then maybe you won't be able to focus so well because these muscles are so tired that they're no longer capable of holding the lens in the particular shape, and you're going to lose the ability to maintain your concentration and focus because these muscles are tired. Now, when you get done and you put your book away because you realize, my eyes are giving me problems, I need to go and relax. So what you do is you relax these muscles. The tension on the suspensory ligaments of the lens then becomes greater because these muscles were pulling it like this. If you relax them, then this all goes back down. And so as the suspensory ligaments pull on the lens, it becomes thinner and flatter, and that allows you to see far away. And notice you didn't have to do anything but relax to see far away. So go outside, look at the clouds, look at the trees, absorb some nature, and these are completely relaxed while you're doing it, and then once they get rejuvenated, you can come back and read. So you need your suspensory ligaments of the lens attached to your ciliary muscle. Well, the ciliary muscle is in the ciliary body. And this area right here, it doesn't really show it that well in this picture, but it actually goes all the way around. and it makes it look like you have this serrated tissue that is related to the ciliary muscle, and that is going to be referred to as the ora serrata. Now, at the base of the ora serrata, you're going to have the beginning of the retina, and the retina is going to be represented in this picture by this yellow nervous tunic. But before we get there, notice that the ciliary muscle and the ciliary body, they are basically continuous with and continue down as the choroid layer of the eye. And the choroid layer of the eye is essentially a very black sheet that sits behind the retina. And the reason it is black and really highly pigmented is the light comes in, it passes through the retina, and some of the light might hit a photoreceptor, but the light that doesn't hit and get absorbed by the photoreceptors is then absorbed by the choroid layer. And so you have this black mat under here to absorb any of the stray photons so that your light is always creating the clearest image possible. Now, you can force light to reflect off the back of your eye. It takes a lot of light to overcome the absorbing capabilities of the choroid layer, but the way you do that is by being in a flash picture. And the flash fires, the light goes in, it bounces back out, and the light that's bouncing out of your choroid layer is captured in the film by the person having red eye. And the reason it's red is because here's all your blood vessels, and they're actually lying very close to the choroid layer, between the choroid layer and the retina. Now, some of them, it looks like here in the optic disc, are above the retina, but if they were above the retina all the time, then they would interfere with your vision. So a lot of them are actually tucked down here under the retina. But the light passes, it hits the choroid layer, it flashes through your vascular system and then out, and now it looks red because it just passed through a whole bunch of red blood cells. Now, if you follow this down, at the margin of the ora serrata, you have your nervous tunic, and this is the retina. And the retina is the receptor. So you had all this other structure so that you could have this receptor. And this receptor is going to have rods and cones in it that are going to detect different wavelengths of light. Now, the cones are capable of producing a color picture. It usually requires a fairly strong light source to do so, but cones are responsible for your color vision. And then rods are basically for clarity. They detect borders, and they also are sort of lightening the image. So rods are black and white, but they help you detect the borders of objects. So like the reason I can see the edge of this picture is because of my rods. In the retina, right here, I have what looks like, if you use an ophthalmoscope and look in somebody's eye, you'll see this area and it will look like a yellow spot. And so we call it the macula lutea, the yellow area. And dead center in the macula lutea, you are going to have the highest concentration of rods and cones, and that is going to be your fovea centralis. So the light should come in, be focused by the cornea, and go into the lens where you can have fine focusing by changing the shape of the lens. And between the cornea and the lens, the light should all come here and focus on the fovea centralis. And that will give you the clearest picture. Now, obviously, there's going to be some light that spreads out in here, and so this is where you have your attention focused, and that's the clearest area. And then this stuff over here that you're seeing would be sort of what's out of the corner of your eye. And if it catches your attention, then there are reflexes that we talked about that would shift this over and cause this part of the picture to be redirected to the fovea centralis. And that is why if you focus on something, it's often called, you're going to foveate on it and study it to make sure that whatever is happening in that area of your environment doesn't mean you harm or whatever. So now we come down here, we're at the back. We've talked about the three layers that form the wall of the eye. And this is the last area that we need to talk about. And this is where we have the connection of the optic nerve, cranial nerve 2. And notice that it's making connection to the receptors, the rods and the cones within the retina. And it's going to carry the sensory information back into the brain. Dead center of the optic nerve, you're going to have an ophthalmic artery and vein. So there is vascularity that is coming in through this area. Now this area is referred to as the optic disc. And in this area we do have some blood supply, but the most important thing is that we have no rods and cones. So this area is incapable of generating an image. And if you come to lab, we'll show you how you can detect your own blind spot, your own anatomical blind spot. But the reason you don't have a dark area floating around in your vision is because your brain is a master of Photoshop, and it's actually going to use information from your contralateral eye to fill in the blank spot that is created by the other eye. And this cranial nerve 2 is going to be covered by dura mater, and then the dura mater is going to transition and fuse and become the sclera around the outer area or outer edge of the globe of the eye. So I think with that discussion, fairly long, I have covered all of the structures that we need to talk about related to the eye. Now obviously there are different types of vision. You have myopia and hyperopia. Those could be defects in the cornea, or they could be defects in the lens, but you should know the difference between myopia and hyperopia. Also presbyopia, you should know the difference between cataracts and where LASIK surgery occurs. So if you can't find that information in your textbook or in the slides somewhere, be sure and ask those during a class. Just for kicks, I'm going to show you this close-up. Here is the cornea coming in. It is the junction between the cornea and the sclera. And notice that this is sort of the anterior chamber, the lateral corner of it, where you're coming in contact with the iris. And notice the iris appears to be somewhat corrugated because you've pulled the iris back and the tissue is sort of bunching up so that you can open the pupil. And then down here we have our posterior chamber. You can see some of the glandular projections, and here's some glandular tissue that is producing the aqueous humor. You can see the suspensory ligaments. They're attached to the ciliary body. And then you have a close-up of the ciliary muscle, and you can see its orientation. So if it contracts, it's going in this direction, and it's causing a slacking in the suspensory ligaments to make it more globular for near focus. And then if you relax this, this all sort of just shifts back down and this becomes thinner. The other important structure here again is the canal of Schlemm, and you can see how it's allowing for the reabsorption of the aqueous humor. But then notice that I have these very small minute channels that go over and are associated with the venous structure. So I get my aqueous humor produced here, flows around, is reabsorbed, and then goes into the venous structures and back into circulation. This picture here is going to give us an idea of how the information picked up by the eyes will then be transmitted to the occipital lobe and to the areas 17, 18, and 19 around the calcarine sulcus. For vision, there is extensive processing of the electrical impulses that are created. Obviously, you have the photoreceptors, the rods and the cones, that are picking up the wavelengths of light and creating the electrical signals. But then those electrical signals are going to be passed to horizontal cells and then finally to bipolar cells. The bipolar cells will then synapse on some ganglion cells, and those impulses will be carried by their axons into the optic nerve. Now the optic nerve is then going to pass toward the brainstem, or on the bottom of the brainstem. And we saw the optic chiasm in several views of the brain. And so here is the key. to not being able to detect your blind spot. So the information on the medial side of the retina, notice that it enters the optic nerve, it passes to the optic chiasm, but then it goes to the contralateral side. The information on the lateral side of the retina stays ipsilateral, and that is true for both eyes. So what's happening is right here, I am able to use the information from this eye to paste over and fill in the lack of information left by the optic disc, and that is true for both eyes. You then go to the optic chiasm. The optic tract will then go down into the lateral geniculate nucleus of the thalamus, and then those will synapse with axons that form the optic radiations, and these are the optic radiations as the information is being transferred to the cerebral cortex to finally create the visual field and the movie of your life that you're watching as you're moving through your environment.